This video is a continuation of topic 7.9, phylogeny. In the previous video, we looked at the tools used to determine the evolutionary relationship between different species. The tools that we talked about include fossil records, morphology, and today we're going to look at molecular evidence, including the DNA and the amino acid sequence. So once again, if you remember from last video, what we saw here is we took a group of species and we sorted them based upon characteristics, and we're able to identify their evolutionary relationship to one another, shown there on the right hand side. So this would be an example of a phylogenetic tree. Molecular data provides the most accurate and reliable evidence, more than morphological traits, in the construction of the phylogenetic tree or cladograms. So in a lot of questions, what you'll be given is a table like you see on the right hand side. You can see the 5 different species there: the horse, the snake, the chicken, the penguin, and the donkey. And then what you'll notice on the top, you will also see the horse, the snake, the chicken, the penguin, and the donkey. What the numbers represent is they represent the amino acid differences in a particular protein between the different species. In this particular example, it's cytochrome C. What you'll notice is that if you compare across the diagonal, the horse and the horse are obviously the same amino acid sequence. The more lower the number is, the more similar those two individuals would be for that particular protein. So for example, the horse and the donkey, there's only one difference between those two amino acids. And as we know, a horse and donkey are evolutionarily very similar to one another. Whereas if we compare the horse and the snake, there's 21 differences between those two proteins. And sure enough, we know the snake and the horse are very different. One is a mammal and one is a reptile. So what we do in this table is we look for the individuals that are the most different. So for example, you can see that the snake is very different from the other ones on our table here. And based upon these differences, this will allow us to build a phylogenetic tree. So here's an example of how this phylogenetic tree could look. As I said before, the snake is the most different for all the different characteristics. You can see that if you compare the horse to the snake, it's 21 amino acids different. The horse and the chicken is 11. The penguin and the horse is 13. So if you remember from before, we would represent the snake as the out group in this example. As we continue, we're going to look at things that are more similar. If you look at the chicken and the penguin, there's only 3 differences between them, suggesting that those two must be very similar to one another. Likewise, the horse and donkey have 1 difference to each other. And so using these similarities and differences, it allows us to build this phylogenetic tree. The other advantage of phylogenetic trees is they can be used to illustrate speciation that has occurred. So these nodes or branch points on the tree represent the most recent common ancestor, and is at that point the 2 groups or lineages separate from one another. So we can see that the common recent ancestor between B and C, at that point the species split into the individual and they evolved independently from that point on. And we'll talk about speciation in the next power. Phylogenetic trees and cladograms can be constructed from morphology similarities of living or fossil species, and from DNA and protein sequence similarities. And this is what we addressed in this presentation already, so you can just further see the examples of this in this chart. Now interestingly, phylogenetic trees and cladograms represent hypotheses. In science, we're always testing hypotheses. Sometimes those hypotheses get revised, and that has happened a few times in the evolutionary trees that we've built from the past. And as we collect more and more data, in particularly the DNA and protein data, it actually forces us to change these phylogenetic trees that we built in the past and basically rearrange them. So you can see this example right here with whales. Whales were once thought to be more closely related to the mesenchymal side. After the discovery of new DNA evidence, paleontologists have actually rearranged these phylogenetic trees, and now hippos and whales are more closely related to one another. So what you can notice here is that these phylogenetic trees can be changed if new evidence supports that change. Here's another question for you. Who is the most common recent ancestor of the dinosaurs? 3 options: lizards and snakes, crocodilians, or birds. Once again, remember that what we're looking at here is morphological similarities, fossil evidence, as well as DNA or protein sequences. Now, based upon what a dinosaur looks like, a lot of us would probably assume that it's either a lizard or a crocodilian. However, what scientists now know is that in fact dinosaurs are more closely related to birds than they are lizards or snakes or crocodilians. So be aware of that. Another question for you is, what kind of organism is this? It looks like a snake, but surprise, surprise, when scientists look at this species, it is actually not a snake. In fact, it is what we call an eastern glass lizard. And what this is interesting is this shows us that legless conditions evolve separately. We call these analogous structures. If you remember that term, I talked earlier about that. So these structures evolved by convergent evolution. And that's something you have to be very careful about. Homologous structures versus analogous structures. Homologous structures are individuals that share a recent common ancestor, and they've diverged from that point. Where analogous structures do not share a common recent ancestor. Instead, they've had the same environmental pressures and used the same solutions to solve those problems. Analogous structures have tricked scientists into building different phylogenetic trees, which we now know are incorrect today. So here's an example you can try on your own. Try drawing a phylogenetic tree based on the data below. You can see the different species that are on the top: the lancelet, the lamprey, the tuna, the salamander, the turtle, the leopard, and the dolphin, and then the different characteristics. Once again, the zeros represent that trait does not exist in that species, and the 1 represents the presence of that. So try drawing a cladogram based upon that information. Here is the answer.